Let's see question number 116 until 120. So first question, the question number 116 says that out of 38 ATP molecules produced per glucose molecule during respiration, okay, 2 are contributed outside glycolysis and 36 during respiratory chain. 2 are contributed from outside mitochondria and 36 inside mitochondria. Then see 2 during glycolysis and 34 during Krebs cycle, and all are formed inside the mitochondria. Okay, so let's discuss. So first thing is that glucose, okay, undergoes glycolytic pathway to generate 2-pyruvic acid, which undergoes complete oxidation inside the mitochondria. Okay, inside the mitochondria, they undergoes. And inside the mitochondria, if you remember, there are 2 pathways or 2 mechanisms running on. One is the Krebs cycle, then the other one is electron transport chain, okay, coupled with oxidative phosphorylation. So here, glycolysis to pyruvic acid conversion, okay, there is an input or investment of 2 ATP, and there is a harvest of 4 ATP. Out of 4 ATP, 2 will be invested again, and 2 will be our gain. Okay, and remaining 36 ATP comes from Krebs cycle, okay, and the electron transport chain coupled with oxidative phosphorylation. Okay, so I will say that 36 ATP come from the mitochondria, inside the mitochondria, and 2 ATP we get from the glycolytic pathway. Okay, so based on this, let's see the statements. The first statement says that 2 are contributed outside the glycolysis. So it's not the outside the glycolysis. Okay, it's from the glycolysis. So it's false. 2 are contributed from outside mitochondria and 36 inside mitochondria. They didn't say from which, but say outside mitochondria, we will take it. Okay, 2 during glycolysis and 34 during Krebs cycle. 2 during glycolysis correct, but 34 during Krebs cycle is not correct because he generated 36 ATP. Okay, so more appropriate answer will be B. Okay, where you 2 ATP you get from the glycolysis. Okay, and 36 ATP you get from the get inside the mitochondria. Okay, that will be 38 ATP. So your option will be option B. Let's go to next question. Question number 170 is from plant physiology. Question number 117 is asked that which of the following hormones prevent ripening of fruit? And the option given are gibberellin, followed by ethylene, followed by cytokinin, and followed by abscisic acid. Okay, so let's see the functions of each of the hormone. Okay, so gibberellin, they are involved in, what are their functions? They are involved in promoting growth and development, including seed germination. They break seed dormancy. Okay, apart from that, stem elongation, delays fruit ripening, and senescence. Okay, ethylene, they promote ethylene, they promote food ripening. It's a gaseous hormone. Then third, cytokinin. It promotes cell division and elongation of plant. Promote cell division and therefore growth. It delays aging in leaves. And D, which is abscisic acid, it's a stress hormone. It has inhibitory effect. Okay. It create dormancy, etc. Okay. So the more appropriate answer, okay, will be the gibberellin. Okay. It can prevent fruit ripening. Okay. So your option will be option A. Question is question number 118. It says that find the incorrect statement. So let's check it. Okay. So which of the following is regarding the growth? We need to specify the growth. So let's see first option A. It says that in plants, growth by cell division is seen only up to certain stage. Okay, it's not correct. Okay, plant has indefinite growth. So it is incorrect. Growth exhibited by non-living object is by accumulation of material on the surface. This is true. A multicellular organism grows by cell division. This is also true. Growth in vitro culture of unicellular organism can be observed by counting the number of cells. This is also true. So you need to find the incorrect statement. So option A is the appropriate answer. Let's check question number 19, 119. So diversity in the type of beaks of finches adapted to different feeding habit on Galapagos Island as observed by Darwin provide evidence for. Okay, as you remember, okay, from the evolution chapter, okay, then Galapagos Island, Darwin went to Galapagos Island, there were different islands and there were finches, okay, with different beak shape, okay, based on their feeding habit. Okay, so if we see option number, first option, that intraspecific variation, okay. So while intraspecific variation do exist, okay, and observed by Darwin, the primary focus of this observation on finch beak was on how these variation attribute to natural selection and speciation. Okay, so it's for natural selection and speciation. If you check the intraspecific competition, okay, so it refers to competition between, or not between, among the species. Okay, among the species. Okay, that means the individual among the species. And while it can drive natural selection, but it does not directly explain the diversity in beak, okay, in types that was observed by the Darwin. Then if we check the interspecific competition, that means the competition between different species, right? So it focuses on the variation within the finch population, how natural selection acted on the variation. Okay, so the best option is origin of species by natural selection. So nature, okay, by selected by nature, the nature helped. The diversity in the type of beaks of finches observed by Darwin, okay, in the Galapagos Island provides evidence of the process of natural selection. So your option D will be more appropriate. Then let's check the next question. The next question is a group of fungi with septate mycelium in which sexual reproduction is either unknown or lacking are classified into. So if you remember mycelium, okay, so there are structure like this, okay. Now this can be septate, okay, they may be septate or maybe they may be aseptate. If they are aseptate, you can see there can be multiple numbers of nucleus inside. Now questions ask about the classification. Okay, so we have Phycomycetes, then you have Deuteromycetes, ascomycetes, and then basidiomycetes. Okay. Now we need to check the septate. Two things: septate mycelium and sexual reproduction. Okay. So let's put mycelium type and reproduction. Okay. So if you check phycomycetes, they are aseptate. Okay, and they reproduce both asexually, okay, and sexually. An example can be rhizopus and mucor. Okay, can be rhizopus and mucor. Then deuteromycetes. Deuteromycetes are septate. They are septate, right? Their sexual reproduction is not known. Yeah, sexual reproduction not known. Right? Then you have ascomycetes. Okay, so ascomycetes, they are septate and they reproduce sexually. They have asci. Okay, right, forming the ascospore. Right, you can take yeast or penicillium. Okay, then you have basidiomycetes. They are also septate. They also show sexual reproduction. Example, you can take mushroom or toadstool. So the question is about the septate and the sexual reproduction not known. Okay, so it is the deuteromycetes. Okay, where you don't see or you don't know the sexual reproduction way, but it has a septate. Okay, so the more appropriate answer is option B. So see you in next video lecture or next solution.